Future genetic technologies will certainly benefit from advances in biochemical engineering. The production of lysine is achieved through a process of fermentation using the bacteria Corynebacterium glutamicum. Production of lysine exceeds 600,000 tons a year. Lysine is an amino acid essential in the nutrition of humans and animals. BASF was able to identify and patent 1,500 genes in the inherited makeup of the Corynebacteria. They use genetic engineering techniques to optimize the metabolism of the bacteria. Proteins that act as scissors, known as restriction enzymes, can be used to cut and manipulate the DNA sequences of the bacteria. This partial sequence forms the genes that control the metabolic processes in the cells. By cutting and replacing sequences, they can force the cells to produce more specific proteins. This process has been used to decode the metabolism of some 200 microorganisms. The process is extremely complex and is handled by laboratory robots. Students are using simulation programs to learn more about metabolic engineering. This is a section of the metabolic network of E. coli. They are learning that there are several ways to manipulate genes to produce certain outcomes. They are researching whether or not these practices are profitable outside of the production of mass market goods. Can these biochemical processes replace chemical processes to efficiently produce mass market goods, such as plastics or solvents? Much of the basis for chemically engineered products is oil or carbon. Many of the ways these elements are processed result in the release of carbon dioxide into our atmosphere, which could contribute to global warming. A possible alternative to oil is photosynthesis. Microorganisms in the form of algae could put us on the right path to cleaner energy production. Oil produced from algae can produce biodiesel. They use an algae bioreactor where nutrient solutions are circulated in a closed container. They must be exposed to the right temperatures and a precise amount of lighting for efficient development. Carbon dioxide is injected into the photobioreactor to trigger photosynthesis. Not only can the algae oil be refined for biodiesel, but it can also be refined into liquid fuels like ethanol and jet fuel. The algae bioreactor plants could be located adjacent to power plants, where the algae could thrive from carbon dioxide byproduct from the power plants.